Good afternoon, everybody, and welcome to Cyrsiau Cadlam Cymru. Welcome to the fourth and final biology session, led again this evening by Mrs. Paula Burt.
Evening, everyone. So this evening, the session I'm going to run is about meiosis and the cell cycle. So just before we get into that, remember the format for the webinar, only to say, if this is your first time looking at these, remember when you get the materials, access to the materials, you'll be able to go through them at your own pace at a time that suits you, ready for any revision you have on this topic. And I will leave short pauses so that there's a natural break for you to attempt things yourself and pause the presentation. So anyway, here we go. So I'm going to split the revision into three areas really, and that is whether you can describe what happens in the stages of meiosis, so that's meiosis 1 and meiosis 2, and we're also going to look as part of that how it is that meiosis generates variation, so it produces cells that show variation, which is really important in terms of natural selection, evolution and survival, although we won't focus on that in this session. And obviously what we need to do, because of quite a frequent exam question, is we're going to have a look at the differences between mitosis and meiosis, and obviously we're going to go over all of that with some past paper questions that I'll walk you through. So let's start then. Now this follows on from last week when we were talking about mitosis, because we talked about interphase, and the cell cycle, even in meiosis, is actually part of it is interphase, okay, and this is exactly the same as mitosis. It's not part of the actual nuclear division, just as in mitosis, but there are standard things that happen in interphase. So for instance, the DNA replicates. Now that's pretty important, isn't it? Because what happens is that the mass of the DNA has to double, because otherwise we won't be able to produce the cells with the required amount of DNA. During this time we get replication of the organelles, and again we talked last week about how it is that we only refer to organelles replicating if they've got their own DNA, so mitochondria and chloroplasts. We also talk about in interphase that organelles are made, and ATP and proteins are made, ribosomal material, obviously very important for making ribosomes. And we say that cells increase in size, but we don't say growth in interphase, because growth is implying one cell makes two, two makes four, etc. So that's interphase, exactly the same as in mitosis. So let's look at how meiosis fits into the life cycle then. Well, this only takes place in the reproductive organs in plants and animals, and it is a process which produces gametes, so sex cells, egg, sperm, pollen, ovule, depending on whether it's animal or plant. And we produce, in this process of meiosis, we produce cells, 4 cells that all are haploid. So that means they have half the number of chromosomes compared to the parent cell that they were formed from in the ovary or the testis or the parts of the plant. So the cells produced show variation, and but as I say, that is not going to be our focus. So I thought I'd just remind you that diploid cells have 2 sets of chromosomes, whereas haploid have just the 1. And this division, even though there are sort of 2 divisions here in meiosis, this process will halve the chromosome number. And that is so that you can see from the diagram I've shown you there, so that after fertilization, where both haploid gametes will fertilize, the chromosome number is restored. So we're talking human number here, so we're talking gametes being 23 and N, and the diploid zygote fertilized egg cell 2N 46. And then of course what happens is that zygote will then divide mitotically in order to grow the organism. So that's just where meiosis fits in the life cycle. Now, also remember, in terms of what happens with chromosomes, we'll get potentially different alleles on chromosomes in male and female sex cells, and so what happens in the cell that's produced the zygote, we'll get pairs of alleles. So this is it, we may have new combinations compared to either of the parent cells. And just a little reminder there that obviously body cells have got 2 of each chromosome, because that's pretty important for the work that we're going to cover. Also, you can see that in both these diagrams, a couple of things here. First of all, we start with a cell here at the top where there's already been DNA replication. So each chromosome is now made of 2 chromatids. And we can see there are 2 stages here. So the first stage is where these chromosomes, which look identical to each other, they've got their centromere in the same place, they're the same shape and size. The only thing that's different is the colour from what you can see at the moment. And so there's a first division where those homologous chromosomes, as they're called, are separated. And in the second division, then the chromatids that form part of each chromosome are separated. So you can see a parent cell of 2 chromosomes produces 4 cells, each with 1. So we've got half the number, haploid number, as opposed to diploid. Now you can see in terms of sort of thinking about human and diploid and haploid number there, you can see that we've got in the first stage, which I'll show you here, going from this orange bubble to the yellow, we get the first division, which actually reduces the number from diploid to haploid, and the second division, again, you know, the haploid number remains, okay? But it's all to do, as I was saying last week, about separation of chromosomes and chromatids. So 2 divisions, haploid number after the first division. So we're going to look at the 2 phases then, and the first thing we're going to look at is prophase. Now, just like in mitosis, what we've got here is you'll begin to, if you look at images, photographs, you'll see that something looks like it's happening in the nucleus. It's no longer a dark mass. There looks like there's strands there. And in fact, what's happening is chromosomes are shortening, they're thickening. And in the case of meiosis, they're finding their homologous pair. That means the chromosome that is the same shape, size, and has the centromere in the same place. I mean, this is different compared to mitosis. That didn't happen in mitosis. So what happens here in meiosis is that the chromosomes associate as homologous pairs, and you can see in the examples I've shown there. So this is called synapsis. Each of the pair, so that's the 4 strands, is a bivalent. Okay, so another name for you. So the bivalent is, I'll show you the diagram down here. So we've got homologous chromosomes. Each one has undergone DNA replication, so we've got 2 chromatids in each, and the structure with its 4 strands is a bivalent. Some people call it tetrad, but you know, a bivalent. Okay, so 4 strands in total. And once again, as I said right at the start of the slide, prophase 1 is the same in mitosis as in meiosis. So all the sorts of things you'd expect, you know, the chromosome shortening and thickening, and the centrioles moving to opposite sides of the opposite poles, sorry, the spindle fibers beginning to develop, the nuclear membrane you can see there in that diagram actually beginning to disintegrate, and of course by the end of the phase it will have gone completely. So that's prophase 1, very similar to mitosis. Now, the other thing to remember in this prophase is there are a few important processes occurring here, and the first thing I want to talk about is crossing over. And you can see from some of the images I'm just sort of putting up for you now, chromosomes that are actually connecting to each other. Okay, so the bivalents that I've told you about, they kind of begin to wrap around each other. Okay, and what happens, some places they repel each other, some places they stay attached, and at the points they stay attached they form something called a chiasma, or chiasmata is the plural. And then at that point where a chiasma forms, there's a swapping of potential swapping anyway. You can see from the diagram down here at the bottom that I'm looking at now, there's like an equivalent part on the other homologous chromosome, and it's possible for them to swap over parts of their homologous chromosomes. But of course, if the alleles on the other homologous chromosome are different, then you end up with these new combinations, as you can see here in this diagram from the letters A and B, and you can also see in the diagram on the left here shown with colours. So you can end up with new combinations. We call them recombinant chromatids, and this is a way in which meiosis produces variation in the daughter cells formed. Okay, moving on from that. The other thing I wanted to tell you about is something called random or independent assortment. If we have a look at these two images here, you can see if we go back to that idea of our bivalents, they are now arranging themselves along the equator. We can see the centrioles radiating some of the spindle fibres, but it really just depends on which way round they arrange themselves, and they arrange themselves very randomly. So here we're just looking at 4 homologous chromosomes. Okay, so what happens in this diagram A, you can see if we just say that the chromosomes that originally came from the egg cell, the maternal chromosomes, are shown in red, and the paternal are shown in black. You can see that when these cells are being formed, they can arrange themselves in an opposite way on the chromosome. So you see this, where the arrows are showing you the chromosomes are the opposite way round. So they actually arrange themselves randomly. So that's pretty important, because that's now going to affect which chromosome is pulled to the poles, particularly, as I say, if crossing over has occurred, you're going to have different pieces of information heading that way to the poles. So we've had crossing over, and we've had random or independent assortment, and so now what happens is we have metaphase, as you can see there. So we've got our bivalents, which are arranged. The structure 4 is arranged either side of the equator. So homologous chromosomes arranged, and as I say, it's been at random. And what will happen now is by the end of metaphase, that spindle will start contracting. And then what happens in anaphase is they're pulled by the centromere. Remember, I said it's led by the centromere to the poles. So this is important, because in anaphase, what's happening is homologous chromosomes are being separated from each other, and that's key to meiosis 1. But just like in mitosis, a really rapid phase, so you don't tend to see a lot of images with this process occurring. Spindle fibers contracting, as I say, and with the homologous chromosomes being pulled to the poles. Okay, so it's really important that homologous chromosomes are separated, and you know that. So what we have now at by the end of anaphase, heading towards telophase, is actually a random mixture of what were originally maternal and what were originally paternal chromosomes. So we're in telophase now, the final stage in the first division. This is the end of meiosis 1, and you can see the standard things that we learnt about last week happening. So the nuclear membrane is going to reform, the chromosomes, have reached the pole. You can see the centrioles there. So what happens now is each nucleus, we started off with 4, now each nucleus has got 2, so we've got the haploid number of chromosomes. Okay, so this is the first division where the chromosome number goes from diploid to haploid. Now, in animal cells, what happens is cytokinesis will occur. It doesn't always happen completely in plants. So I just thought, again, I would just re-emphasize this idea of the 2 divisions and diploid to haploid in the first division. So in meiosis 2, this is where we actually separate sister chromatids. So now we've got prophase 2. So again, as in prophase 1, we've got all the usual things happening there. Okay. There's a slight difference in the way the spindle is formed, because now the spindle forms at a right angle compared to where the first spindle formed. Okay, so if you look at some diagrams, you can see that. And as I say, in plant cells, a new spindle isn't needed because the original one is still in place. There hasn't been like a full cytokinesis occurred there. So we've now got the original cell, which is composed either completely 2 separate cells or looks like it's made of 2 separate cells, as you can see in the image there. And in metaphase, what happens then is chromosomes lining up on the equator, and the chromosomes, you can see they're attached to the spindle again by the centromere, and as a metaphase is followed by anaphase, you can see that the this time the chromatids are separated, pulled to the poles by the centromere, and you can see in the little image I'm showing you there that actually that one original cell has got 4 things happening. That's how you can recognize that this is meiosis 2. Okay, and then telophase, final phase, chromatids are uncoiling again, they're lengthening. What's happening now is that the nuclear membrane is going to begin to reform, cytokinesis is going to form completely, and you end up with 4 haploid gametes. Okay, and the thing is though that because we've had crossing over and we've had random and independent assortment, we have now got genetic variation. So let's look at some little images just so that you're reminded of how to recognise the different phases, because we've just got sort of 4 images here and I've focused very much on the second stage. So you could tell this would be prophase 2 because there are sort of, you know, 2 parts. It looks like there are 2 cells and there's like a faint suggestion that there is one overall. You see outside there. So there's prophase 2. You can tell it's prophase 2 because you can see there's strands in there. Metaphase 2 again, 2 parts of cells meeting at the middle. Anaphase again, after, as I was talking about last week, being pulled to the poles, and then you can see in telophase there's very definite the chromosomes have uncoiled again. You can't see the membrane having formed here, but it's very much a sort of a tight nucleus looking there. So I thought I'd just sort of bring some images back to remind you how it is that you can recognise the different phases. Variation, I know I talked about it before, but just again to summarise, it's crossing over in prophase 1. That is really important because chiasmata will form in that case, and then also you have the random alignment and then the separating, the independent assortment of the maternal and paternal chromosomes during the metaphase 1. Now, it's not part of meiosis, but another way in which variation is generated with gametes is that gametes are different from each other. So just random fertilization of different gametes will also generate variation. So again, it's another way in which you get variation as a result of producing gametes, but it is not part of meiosis. And in terms of comparing mitosis and meiosis, I've just summarised it for you on a table there because it is a very common little question in tests and so on. 1 division as opposed to 2. We start with 1 cell, we produce 2 cells in mitosis, but 4 cells in meiosis. We go from diploid cells to haploid in meiosis, but we stay with the diploid number in mitosis. We know in meiosis we're generating genetically different haploid cells, but it is very important for growth, repair and replacement in mitosis that they remain identical, as we talked about last week. Meiosis, crossing over occurs, chiasmata formed, and there is no such thing in mitosis. And segregation or separation of the homologous chromosomes takes place, and the pairing of homologous chromosomes takes place in meiosis, but none of that occurs in mitosis. So here we go. Here's the sort of overall summary of haploid, diploid, 2 divisions. So I'm hoping now, you know, with some of these images that you're a lot more confident and you're recapped. So here we go then. We now need to look at some past paper questions. So what I'll do, I'll read through the question and then I'll give you a little bit of time. I'll give you a pause where you can then decide on the answers for yourself, and then I'll reveal them. So first question for this session then. Complete the table to show whether each role applies to mitosis, meiosis or both. If the role applies, put a tick and a cross where it doesn't apply. So really the question isn't asking for a yes or a no. You know, this is where you need to look at the question and follow the advice they're giving you. Put a tick and a cross. So just take a moment to decide which of those apply to mitosis, which apply to meiosis? So I should just pause now before revealing the answer. Okay, so now that you've had a moment to think and to pause, those that are involved in growth. So let's have a little look. Mitosis, yes, definitely. Growth, repair and replacement, as I've said earlier on. Is meiosis involved in growth? The answer is no, it's not, is it? It's involved, you know, in another process, which I won't give away at the moment. Does it produce variation? Mitosis? And the answer is no, it produces genetically identical daughter cells. Whereas meiosis, does it produce variation? Yes, it certainly does, through crossing over and random alignment, independent assortment. What produces haploid cells? Is it mitosis? Absolutely not. It's diploid cells. Meiosis? Yes, definitely haploid cells. And which of these processes occur in plants? Yes, definitely mitosis. Meiosis? Yes. I mentioned at the start about the fact that the gamete cells produced in meiosis are egg and sperm if it's animal, but also pollen and ovule if it's plant. So yeah, all of these processes are occurring in plants too. So let's look at another question. Now, very often questions require you to draw something in. So I've picked a question now where we can have a look at how it is that we go about that. So it says the photograph shows the pairs of chromosomes found in a body cell of a mouse. And we can see there we've got an X and Y chromosome pair, so we know this is a male. We've got, that's the 20th pair, so we've got 40 there in total. And the question then goes on to ask, what is the diploid number of the mouse? Okay, so the diploid number, remember, is the body cell number. So have a little think about your answer. And your answer should be 40. And it would be very easy to say 20 pairs there, but you're not really answering the question, are you? Because it asks about the diploid number, and that number is 40. So just have a little think about that. Then it says the chromosomes in pairs 1 and 19, if you have a look at that, so pair 1 here, pair 19 here, just before the sex chromosomes, are commonly represented diagrammatically as. So we can see there, you know, they're not identical, but you know, they're not identical to the photograph, but you know, we can recognize them all the same. Now, I used this question last week, but I used it in terms of mitosis. So this time I'm actually revealing the real question, which is a question about meiosis, because it says, using the cell outline below, draw diagrams to show how these pairs of chromosomes are arranged in metaphase 1 of meiosis. Now I suggest, and again I will pause, I suggest you think about the equator is there, so you'll know where the spindles are, you'll know where the centrioles are. How would you arrange those? Remember, it's metaphase 1. So remember I said the two things that happen basically in the two phases. In the first phase, we get separation of homologous chromosomes, and in the second phase, we get separation of the chromatids from chromosomes. So have a little think how it is that you would arrange those. So I'll just pause. Okay, so I'm hoping now I've left you a little time to think about this. So let's start to answer. So there we go. What I've done is I've arranged the first pair, and you'll notice from what I said before, the bivalents are either side of the equator. Okay, so we've got the 4 strands, the bivalent, one homologous chromosome on one side of the equator and the other one on the other side. So let's go now for chromosome 19, and you can see the same thing happening there. Okay, so that is how you would draw those. Now, It is. I mean, it says in the question there that actually you need to draw diagrams, but actually it doesn't say about labeling. There are other questions you'll see that will say label and label. And sometimes you might think, well, what have I got to label altogether? Well, label as much as you can accurately. So, you know, you've got an equator, you might have a spindle, you might have centrioles. You can certainly see in these are homologous chromosomes, or the two together is the bivalent. You've got chromatids, you've got a centromere. There's lots and lots of things you could possibly label with accuracy there. So it's important. Look at the number of marks and try and judge how much detail that you'll need to actually provide. Okay. And then just one final thing. Remember, I said this is the phase. I was trying to give you a bit of a clue that actually this is the phase where homologous chromosomes are separated. So homologous chromosomes are going to be lined up either side of that equator to be separated, obviously after metaphase 1 in anaphase 1. Okay, so brilliant if you've got that correct. Now let's move on. So the next part of the question, it says, using the cell outlines below, draw diagrams to show how chromosomes would subsequently be arranged in metaphase 2 of meiosis. Now, I've given you a little bit too much of a clue here, because what I've realised is that I've actually already drawn them in for you. But let's go over the main points of what I've done there. OK, so I've said that there's the equator. I've drawn that in a little bit more clearly so you can see. Now, if you go back to the previous diagram, you can see what I'd said about the spindle fibre now being at 90 degrees. So the equator before was vertical, now it's horizontal. So actually, we're now in that second phase of meiosis, and in metaphase 2, again, we're meeting on the equator. Okay, so the spindle's at right angles from where it was. The centromeres are attached to the dotted line. That's really important. You know, your accuracy with things like this, it's very easy to think, oh yeah, that chromosome looks just about right. Please be as accurate as you can. You know the centromeres are attached to the spindle fibres at the equator, so make certain you show it. It's obvious. You know, from you to the person who's marking your work, exactly what you're showing there. OK, and again, what's happening? The chromosomes again can randomly align. So I've shown them like this. In this right hand cell over here, it might be that the little one, chromosome 19, was in this position and chromosome 1 was in this position. They are randomly aligning. OK, but because this is metaphase 2, we're looking now at separating those chromatids. So we can separate them, but obviously not in metaphase 2, in anaphase 2. And then what we're looking at the last part of the question there is 3 ways in which meiosis contributes to variation. And I didn't give you much of a time to pause there, sorry, but actually with all the things we've said before, independent assortment, crossing over in prophase 1, it's really important that you mention these things. Prophase 1 is a really important phase, for crossing over. Okay, independent assortment can occur in terms of homologous chromosomes in meiosis 1 or 2, but crossing over just occurs in prophase 1. And haploid gamete cells will therefore show variation. Okay. So I think that's just about that question done. Let's have a little look at this next one. So what I'm going to do here a second is I'm just going to alter this a second so that I can just talk about this gradually for some reason. There we are, that's better. So what we've got here now, another question. So mitosis and meiosis, it says, are both forms of nuclear division. Both types of nuclear division are involved in the production of gametes. Image 1 below shows a section through part of an anther of Lilium species, showing stages in the production of pollen grains. So we can see there it's pointing to A, which looks like it has two halves, maybe of the two parts to that cell. B, it's pointing to a structure that maybe it looks like it's got four parts to it, and C is pointed to some cells around that. And it says there, it was concluded that structure A had just completed meiosis 1 and structure B had just completed meiosis 2. Explain how you would draw these conclusions from the photomicrograph. Okay, so I'll just pause and you think about what I'd said when we looked at the images earlier on. Okay, so now here we go. I'd said about them comprising of two cells. You can see the sort of the two nuclei there, okay, from the one division, okay, and whereas B appears to comprise, to be made of, four nuclei there, so that's formed from two divisions, okay, and you could see that. That's why I was drawing your attention to those images earlier on. And then the last question here on this page says, the cells labelled C are an interphase. Explain why nuclei are only visible in some cells and where nuclei are visible, why they appear to be of different sizes. So this isn't a question strictly about mitosis or meiosis per se, but it's a question that very often comes with cell work, for instance, okay? So, and it's all about sectioning. I mean, you might think, oh, if it's interphase, they can ask me a question why it is that those cells are much more common. And you can certainly see around the edges of that photograph there are more cells in interphase than in the other phases, because it's the longest phase, as we discussed last time. But this one today is just about the way you section things, okay? So if you just imagine a nucleus, or this structure as a sphere, it just really depends on where you actually take your section, where you slice through. Because if I said at the bottom there, if you think the nucleus is a ball, if the section is taken through the top, it's going to be a smaller slice than at the bottom. Sometimes you will cut through the nucleus, sometimes you may not, if it's a whole cell. But if you're cutting through a nucleus, sometimes it'll appear different shapes and different sizes because of the sectioning. Okay, so that's that question done. Okay, this next question. A drawing was made of an animal cell during anaphase. The diploid number in this cell is 8. Okay, so that's pretty important piece of information there, the diploid number, okay? Now we can see there we've got 1, 2, 3, 4, 5, 6, 7, 8 strands, okay, and we can see each of those strands, single strands, being separated from each other. And it says, deduce whether this cell is undergoing anaphase of mitosis, anaphase 1 of meiosis, or anaphase 2 of meiosis, and explain your answer. Okay, so let's have a little think about that. Now, the reason that this can't be mitosis is because it tells us that vital bit of information at the top there. The diploid number is 8, okay? And you can see what's heading to the poles here are 2 lots of 4 chromosomes, as they'll be properly known as when they get to the poles. So this can't be mitosis, it has to be meiosis. So now we need to decide, is it anaphase 1 or anaphase 2? So I'll just give you a little pause and then I'll reveal my answer. Okay, so here we go. My answer is that in anaphase 1, homologous chromosomes are separated, but in anaphase 2, chromatids are separated. And you can see there, it's not a double-stranded structure that's being separated from another, right? You haven't got your bivalents there being separated, the homologous chromosomes being separated. You've literally got 2 separate strands, which must be the chromatids in a chromosome being separated. And so that is why this has got to be anaphase 2. So I'm hoping that makes a bit of sense to you. Okay, so next question, on we go. So the photograph, it says, shows the chromosomes from a blood cell of a mammal. Explain why red blood cells could not have been used to produce the photograph of the chromosome. Okay, so let's have a look at things here. Now, if we think about red blood cells, we know red blood cells don't have a nucleus. So actually that is something that we've been learning about for years, really. They couldn't have come from the red blood cells because we haven't got a nucleus there filled with the chromosomes. The next question. Use the photograph to deduce the sex of the mammal and explain your choice. Now have a close look at the number of chromosomes, whether you can recognise any pairs, and whether, because there are, if you look at them, there are 6, do you recognise 3 identical pairs? So have a little look at that. Okay, so I'm just going to pause. Okay. So what I've said for the second answer is you can see there are 6 chromosomes in total, but there's only 2 matching pairs. So let's have a little look. This one here is the same as there. So that's an homologous pair. Then let's have a look, because this one's quite a large one. That one's quite a small one. These 2, again, matching pairs, homologous pairs. And so we're left with the 5th and 6th, which is this large one, this little one. And so as we know from what we've learned before, that actually the Y chromosome in this case is smaller. So this is likely to be, well, this will be a male because we've got an X and a Y chromosome. So in that case, you have to look carefully, because a quick little scan of that and you just think, yeah, they all look fairly similar, but they're not. And they're definitely matching pairs. Okay. And then the next question says, in this mammal, how many chromosomes would be present in a kidney cell and gametes? So this is asking you a question about body cell number and gamete cell number. So just a pause and reveal. The answer will be 6, which will be the diploid number, and 3, which will be the haploid number. So that's simply a question about that. And again, because examiners and us teachers love asking you questions about sort of drawing in chromosomes, I've included another one, and it says the circle below represents the outline of the mammal cell at metaphase 1 of meiosis. The dotted line is the equator. Complete the drawing to show the spindle and how the sex chromosomes as shown above would be arranged. So slight little variation on things now, so I'll pause again. Okay, now I'm going to reveal. Here we go. So we've got, they're not homologous chromosomes as we're used to seeing them, but they are the pair. So they would be arranged either side of the equator. But it does say show the spindle. So now I'm actually popping the spindle in as well. And please don't forget things like that, okay? So this is metaphase 1, the homologous chromosomes are lining up either side of the equator. That happens with the sex chromosomes too. And please don't forget to draw those spindle fibres. Now, just moving on to some other questions, explain the importance of meiosis in an animal, 3 marks. Again, what I'm going to do is I'm going to give you opportunity to pause, but I've already explained this, so I'll probably move on quickly. So I'll pause and then I'll just reveal the answer. So we know meiosis produces haploid gametes. The diploid number is restored after fertilization because we don't want doubling to happen after fertilization. We've got genetically different cells, and that means we've got variation in the offspring. And another question, there's a range of questions here that I thought I'd show you. And a question on the left actually gives you diagrams. It's a QER question. Basically 3 parts to that question if you look at it. The one part is about, it's asking you about mitosis. The other which is over here. The other ones, you know, part of the question is about meiosis, which is diagram A, and then it's asking you to compare mitosis with meiosis, and this is bringing in that uncontrolled cell division that we talked about last time as well. But there are other questions here that you can challenge yourself with and have a little go at. Just another example of maybe 3 parts to the question. So here we go, QER questions. Don't forget, if you come across that, look for those 3 parts, because remember, you have to actually answer on all 3 parts. So I'm just giving you some details there of, you know, how you would chunk up the questions. You do your keyword stack, as I've told you before, get your keywords together, put them in sentences, put them in a sensible order, because then you hit all the marking points, which are, you know, you've got an articulated sequential answer that fully addresses that question without any major omissions. And as usual, what I'm going to do is I'm going to leave you with 2 things. I'm actually showing you now a series of keywords which you can put into sentences, just like I mentioned would be the approach to the QER question. Here they are, and when they've all, there we are. When I'm just going to say, when I've completed showing you, then again, what you do with those questions is you will put them in sentences to answer the question. And as I'd said to you last time, there's the dice sheet there. This one actually talks about you know, mitosis, meiosis and mitosis, and actually, as I've said to you, you can spend some time writing your own answers and then pause or come back to the answers that I've given you as well. So there we go. That completes our catch-up session for meiosis in the cell cycle. And I just want to say thank you very much for sticking with us. I hope you found the sessions really helpful and informative. And I just want to say keep a lookout for more sessions at some point from Catch-up Wales. So all the best with your tests and your exams. Good luck, everybody.